Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillahirrahmanirrahim. Wassalatu wassalamu ala asyrafil anbiya'i wal mursalin wa ala alihi washabihi ajma'in. Rabbi shrah li sadri wa yassir li amri wahlul 'uqdatam min lisani yafqahu qawli. Amma ba'd. Assalamualaikum warahmatullahi and very good day to everyone. Right. Alhamdulillah, we meet again. Let's continue our class with we're still in the same chapter, which is chapter 5, DNA hybridizations. But let's continue our second methods that's still using hybridizations. For the last 2 weeks, we learned about sortem blots, which is we learned how DNA and DNA interact in sortem blots by using hybridizations. And today we go to the second method, which is DNA microarray. Okay, so DNA microarray. So this is basically the how we call it, the instruments to detect DNA in the chips in the microarray, right, using a laser. And the definitions, this is the definition, the terms that you guys should know regards on the DNA microarray, which is the first one is a target. So target basically is the nucleic acid cDNA sample who identified and quantified have been measured. So the target itself is a sample. The sample that you want to use, the sample that you want to elucidate the contents of the DNA in the target sample. And the second one is fluorophore. Fluorophore is actually usually is a green and red labels attached to the target to enable visualizing expression. Means that if you have the target, so the target should be in a cDNA form, and the cDNA sample will be attached with the fluorophore, right? Basically either greens or reds. So usually red they use for a sample, a virulent sample, for example, to be tested. But then green is basically used for a control. Okay, so at the end, when they want to analyze, the color will be appear, whether in greens, reds, or combinations of green and red become yellow lah. Then I will show you in this slide later on. Right, the third one is microarray. So microarray actually works as a reverse hybridization method, converting from mRNA to cDNA 3 to 5 prime with TTTN. Right, so basically the sample that we use is in mRNA form. So from the sample, the cell, we extract out the mRNA. So mRNA, as you guys learned during transcriptions, so from DNA will be transcribed into mRNA, and from mRNA will be translated into the protein. So we use mRNA as a sample. Okay, we extract out the mRNA from the sample, and from the mRNA we cannot directly use for DNA microarray. Instead of using it directly, we have to reverse transcriptase, right? So we have to use reverse transcriptase to do a reverse replications. Okay, so from mRNA, reverse transcriptase will help to transcribe to the cDNA, which is legs of introns. Okay, so we become 3 to 5. So mRNA is basically from 5 to 3 because in the cell it will be transcribed from 3 to 5. That mRNA is 5 from 3. And when it want to be reverse transcriptase, we will transcribe from 5 prime to 3 prime, which is terbalik lah, 3 to 5. And the last one, the same that we use in sortem blot, which is prop. So prop is an attached nucleic acid with a known sequence, or we call it DNA chips. So prop basically is like an oligonucleotide. Okay, oligonucleotide. In Sorten blot case, we labeled the prop, right, with 32p phosphate, the active elements. But then in the DNA microarray, we use fluorophore. We use fluorophore that bind to the target, right? So it's different. Dalam Sorten blot, prop oligonucleotide kita design tu kita attachkan dengan the detection marker, which is fluorescence. Not fluorescent. Fluorescent also can, but mainly P22 active element. But then in this DNA microarray, we use target. So target tu kita bindkan dengan fluorophore, which is either green or red. Okay, so this is the example of DNA microarray slide sample. So if you can see, this is like normal slide that we use for microscope. But then in the middle, right, actually there is how to say, a grave. It's like macam ada kawah, small kawah lah. So the small kawah appear to be filled by the mRNA and the prop. So they will be hybridized in the well, and it will give color like this after it be treated with the laser lah. Okay, so if you can see there is a lot of a dot color, greens and red and black color. Okay. Right, so let's go to the basic step of microarray. First, you have to obtain the cell with genes that are needed for analysis. Okay, so the advantage of this microarray, you can run 1,000 sample in one experiment, right? So you want to detect a lot of samples, all right? You want to detect a lot of genes, so you can use the DNA microarray. So first, you need to obtain a cell with the gene that you want to use for the analysis. And the second one, you have to isolate the mRNA using an extraction buffer, right? Then you have to remove the buffer with the RNA. Then the mRNA will be 5 to 3 prime, as I said before. The mRNA itself is from 5 prime to 3 prime because it be transcribed from the DNA from 3 prime to 5 prime with the repeating AA tail, right? Poly A tail in mRNA. So as you remember, the mRNA will become matured if there is a poly tail A in the 3 prime, and then in front there is a cap. So when both is appear in one mRNA, it become a matured mRNA. And then you need to convert each mRNA into colored cDNA, right? So if I mentioned, the mRNA need to be using reverse transcriptase to do reverse translations. Okay, then the translated cDNA will be converted into color cDNA, right? So the target labeled with fluorophore, either in green or red color. Right, next, with the mRNA degrade reverse cDNA created, mixed color sample of the cDNA. This one that I explained before just now lah. Okay. Right, so this is the microarray or DNA chips on the slides. Okay, if I show you this picture just now, right, in the middle of the slide there is like a cloudy colored area, which is consists of thousands of very small well. Okay, so this is actually the small well on the slide, right? Every spot on the chips represent a different coding sequence from different gene. So this is meaning saying that you can, in DNA microarray, you can run 1,000 experiment or 1,000 different gene in one experiment. Each spot on the chips is made of a DNA prop that can pair with the cDNA that was created. So inside the well, actually, there is already have a prop. So you already designed the prop, the oligonucleotide, a specific oligonucleotide that can bind directly to your sample, which is cDNA. So prop dari dalam ni lah, dalam well ni, right? So It's just a matter of you put your DNA into each well, and the probe will be hybridized with the cDNA. And if it hybridized, it will give a color. Then you can observe it. Right. Then incubation with the mixed cDNA and the chip DNA with yield some pairing. Wash off unbound cDNA to see what has bound to the microarray. So when there is a probe in the well, all right, on the DNA chips, then you put your DNA sample, cDNA sample, and you incubate to give a time for the cDNA and the probe to be hybridized together, right? So if there is a complement, the probe will bind to the cDNA tightly. But then, if both are not complement, your cDNA cannot bind to the probe, and the unbound one will be washed away during a washing step. Right. Then, after incubate, after you wash, now you will visualize the bound cDNA. The slide with the microarray chip is placed inside a dark box where it scans with the high resolution laser that detects the bound fluorescent label. So basically, the cDNA tadi you dah attachkan dengan fluoropore kan, either green or red color. So this green or red color can be detected using high resolution laser. The information and image are then sent to the computer for analyze. So they akan analyze lah, right? And it gives like sort of like this, okay? Creates a ratio image, green image signal expressed in one condition, red image signal expressed in one condition, yellow image signal expressed in both conditions. So means that usually green color ni is a control sample, okay? In the red color is appear to be a virulent factor lah, yang tadi kalau you nak tahu your target then cDNA. But then in yellow color ni both boleh bind lah, right? Okay, so this is the summary. I put a summary slide for the DNA microarray. So for example, you have a cell, right? So this is the sample that you want to be tested. And this is a non-virulent, a reference. This is a virulent. And first foremost, you need to isolate the mRNA. So you need the isolations of mRNA with polyethyl A. And so this is the polyethyl A, considered as mRNA. And you need to complement DNA synthesis using reverse transcriptase. So this is 5 from 3. So 5 to 3, 5 to 3. And the reverse transcriptase will be transcribed from 3 to 5. With oligo-DT. So this is TTT lah, complement for the polyethyl A. Right. So after finish reverse transcriptase, the cDNA created will be labeled with the fluorescence of fluorophore, as I mentioned just now. And for virulence, it will give red. And for non-virulent, a control reference is on a green one lah. Okay, after you hybridize with the target probe, right? So the red color saying that there's appear to be a virulence factor from the cell. But then from the green color, it's nothing lah. There is no virulence lah, because it's a control. And there is the yellow one, right? Unchanged genes, constitutive expression, means that both can be present. Right, then the sample will be scanning, all right, will be analyzed by computer, and you give a result like this one, right? Right, so the capacities of the DNA array. So this is the advantage of DNA array lah. Not limited to human genetic material. So can be used for all species. It doesn't matter you nak pakai kat human ke, you nak pakai kat plants ke, you nak pakai kat bacteria or yeast, you can use DNA microarray as long as you have a large quantity of samples, right? And you want to detect a specific gene portions in the very long DNA. Can be displayed thousands of different genes, pending chip size, right? 100 nucleotides, 100,000 nucleotide. This is the chip size lah, right? Allow the study of multiple genes at once. So this is the advantage of the DNA microarray. And let's see the applications of microarray. Right? First, identify genes and the gene mutations of the different types of diseases such as cancer. Okay, so you can detect is there any presence of cancer genes or cancer marker in the cell, right, by using DNA microarray, and identify the expression level or quantity of the gene mutated or not mutated. So if there is a mutations in the gene, for example, due to the UV radiations or alkaline agent that can cause nucleotide sequence is mismatch, right? So it can be identified using DNA microarray. And the last one is fish fluorescence in situ hybridization. So fish, DNA microarray, and southern blot. So these three methods using the same principle, which is DNA hybridizations, but the method is a little bit different, right? And the last one is fish. So fish is it's a short form of fluorescence in situ hybridizations. In situ means that you do it at this site. So you do it at that particular site, right? You didn't extract it out, just put it directly into the cell, and the cell will be, the prop will be hybridized with the target cell, and it will give fluorescence in situ. So you can observe in situ in situ place. Okay, so fish is a unique DNA fragment specific for a single chromosome is labeled with the fluorochromes, which may be visualized under ultraviolets, right? So this is basically almost the same as a DNA microarray just now. A hybridization reacts allow the labeled DNA fragments to bind to the complementary DNA in interface nuclei or metaphase chromosome. So there is two rules that you need to follow, right? So first one, the hybridization reactions allow the labeled DNA fragment to bind to the complementary DNA. So the labeled DNA fragment is the prop, right? Or known as a prop, oligonucleotide prop. So it's only allow the props to bind the complementary DNA in the interface of nuclei. So interface is not interface is consists of S phase, G1 phase or G2 phase. So in M phase, the binding, the hybridization will not be occur. So it only occur during interface, either S phase, G1 phase or G2 phase. Or it can also in metaphase chromosome, right? Sometimes it can be in metaphase chromosome. And the DNA fragment can be visualized under a UV microscope, and signal of different color can be identified. So it's like this. So you have a cell, a plate of cells, okay? Then you want to observe in situ reactions, which is you have to put your oligonucleotide sequence or prop, labeled prop. The prop is been labeled by the fluorochromes or fluorescence. So the specific prop will bind to the specific sites in the DNA, right? Then after incubation, after washing, so on and so forth, you can directly observe in the cell using UV microscope. So if in Southern blot, you can extract out the DNA. In DNA microarray, you have to extract out the mRNA. But for fish, you don't need to extract out. You directly put the prop on the cell, and will bind to direct target, specific target in the DNA. So this is example of the fish analysis. Okay, so if you can see here, all right. So this is the nucleus of the cell. I think this is HEC293 cell. Okay, so this is nucleus. The blue color is the nucleus. So in the nucleus, of course, there is a DNA chromosome. in the gene. And when you already put a probe and incubate it for a while, and you observe under UV microscope, you can see there is a dot appear green color. So this is example of the fish, means that the probe successfully bind to the target DNA, right? And it will give a color, fluorescent color, right? So this is the fluorescent color. So it means that this is your target gene, and this is your target gene site, right? And this is the control lah, right? Control. So there is no fluorescent, all right? And yeah, this is also example of the fish. This is a positive one. So this is the control. Right. So I think that's it. We stop until here. Then on next class, we will continue chapter number 6. We will continue after Raya break lah. Right. So next week is Raya break. So you have your Raya. And after your Raya, we'll continue with chapter 6. And I didn't mark your test 1 yet because I have to mark around like 134 paper. Means I have 134 students from A2 until D1. So it will take some time. Then I promise you, once I finish marking your test 1, I will let you know the markah lah, the grade. Each of you. Okay, so if you have any questions on DNA hybridizations chapter, you can freely to ask me through the WhatsApps, or you can left your questions in the comments columns in the Google Classrooms. Then I will answer it as soon as possible. Okay, alright guys, thank you so much. Jangan lupa niat puasa esok. Assalamualaikum warahmatullahi wabarakatuh.